Okay, let's dive right into this explainer. Today, we're unpacking how Google DeepMind completely shattered biology's most notorious puzzle. For decades, scientists have been totally stumped by a biological enigma sitting right at the foundation of life itself. But in a stunning leap forward, artificial intelligence didn't just help solve this puzzle, it cracked it wide open.
So, this is the 50-year mystery of protein folding. It's all about understanding how a linear string of amino acids magically folds itself into the complex 3D shapes that operate all living organisms. Think about it. Your DNA is essentially just a one-dimensional blueprint. It's a linear string of code. But living, breathing organisms are 3D. How in the world does a flat linear string of code know exactly how to crumple, twist, and fold itself into a highly specific functional machine? To put things into perspective and anchor why this matters, historically, it cost up to $1 million and took years of grueling experimental work just to map a single protein structure. Scientists had to use incredibly complex, tedious lab methods. Imagine trying to develop a life-saving drug, but you have to spend $1 million and wait years just to see the shape of the biological lock you're trying to build a key for. We're going to treat this explainer like a tour through a futuristic assembly line, breaking down exactly how AlphaFold 2 achieved this Nobel Prize-winning feat. We'll cover 1. Biology's 50-year mystery, 2. The evolutionary cheat code, 3. Inside the Evoformer engine, 4. Building the 3D structure, and 5. The final physical polish. All right, part 1. Biology's 50-year mystery, the holy grail of science. Let's start with the sheer scale of the problem scientists were facing. The crucial takeaway here is that DNA only gives us the 1D blueprint. Life simply doesn't function until that blueprint folds into a perfectly aligned 3D machine. Proteins orchestrate absolutely everything in your body. From the antibodies fighting off a cold to the enzymes digesting your lunch, they all rely on their physical shape to work. If a protein misfolds, it doesn't function, which unfortunately is the root cause of many severe diseases.
Decades of frustration literally vanished in 2020. And AI suddenly started generating remarkably accurate predictions in seconds, completely rewriting modern biology. What used to take years of manual labor was suddenly happening instantly. The breakthrough was so profound that it actually earned the co-creators the 2024 Nobel Prize in Chemistry. But how exactly did they pull it off?
Which brings us to part 2, the evolutionary cheat code, reading the clues. Instead of trying to calculate quantum physics from scratch, AlphaFold acts more like a detective, looking for evolutionary clues left behind at the scene. By searching databases like Uniprot and the Big Fantastic Database, and yes, that is absolutely what it's called, AlphaFold stacks up sequences to find the evolutionary fingerprints of how these molecular strings have changed over billions of years. Think of a protein's amino acid sequence like a string of text. Nature has spent billions of years optimizing these strings. AlphaFold lines up a target sequence against millions of others from evolutionary history, creating what we call a multiple sequence alignment. For example, hemoglobin in a human and hemoglobin in a horse do the exact same job. Because of that, they share a very similar structure, even if a few of the building blocks have mutated over time. By comparing similar proteins across different species side by side, you can easily spot specific amino acids that seem to mutate in tandem. This covariation analysis is a massive statistical cheat code. It gives the AI its first major clue about the physical layout of the protein. If we notice that whenever amino acid number 5 mutates, amino acid number 120 always mutates right along with it across all these different species, well, that's a huge red flag for the AI. It tells the system, hey, these two parts of the string must be physically bonded to each other in 3D space.
Now let's step inside part 3, inside the Evoformer engine, the dual processing core. This is where the heavy lifting happens.
The Evoformer continuously refines two vital tables at the exact same time, allowing information to flow back and forth between evolutionary clues and physical geometry, using specialized self-attention mechanisms. It's essentially managing two massive spreadsheets simultaneously, one tracking the evolutionary history we just covered, and a spatial matrix tracking the physical distance and orientation between every single pair of amino acids in the sequence. To force the neural network to respect real-world physical space boundaries, the Evoformer uses a basic geometric rule, the triangle inequality theorem. AI can sometimes hallucinate, but the physical world has strict rules. By applying a triangle multiplicative update, the AI enforces rigid mathematical logic onto its structural guesses.
Through these steps, the AI ensures that if two parts of the protein are far away in the math, they cannot impossibly overlap in physical space. Think about it. If point A is right near point B, and point B is near point C, then point A cannot possibly be miles away from point C. Checking these triplets of residues guarantees the distances actually comply with reality. It forces the model to be physically realistic.
Let's see how this abstract math builds into a physical molecule in Part 4, Building the 3D Structure, High-Tech Origami.
To create actual 3D Cartesian coordinates, meaning exact X, Y, and Z points for every atom, AlphaFold simplifies things. It treats each amino acid as a rigid puzzle piece with a flexible arm. It imagines the peptide backbone as a free-floating triangle in space that can move up, down, left, right, and rotate freely in any direction.
Now, traditional AI models get completely confused if you just rotate an object. So AlphaFold uses this custom math called invariant point attention, or IPA. It ensures our high-tech origami folds correctly regardless of its orientation on the virtual canvas. If you turn a protein upside down, the atoms are still connected the exact same way, right? IPA allows AlphaFold to stitch the backbone and side chains together perfectly no matter how the molecule is shifted in the simulation.
And the best part? It doesn't just guess once and quit. This recycling process allows the model to double-check its own work, pulling the atoms into tighter, more accurate alignments with each pass. It takes its best 3D prediction, feeds it right back to itself, and asks, can we make this even better? It does this up to 3 times to find the absolute lowest energy, most natural shape.
Finally, our AI-generated molecule reaches the end of the assembly line for Part 5, the final physical polish, OpenMM relaxation. Because AI can sometimes generate microscopic physical impossibilities, like 2 atoms occupying the exact same space, OpenML comes in to ground the mathematical prediction in hard physical reality. The neural network did an incredible job getting us 99% of the way there, but we need standard physics to act as a kind of molecular chiropractor and carry us over the finish line. This rapid cleanup phase shifts the atoms just enough to relieve any chemical tension, handing scientists a pristine, workable 3D model. It performs what's called energy minimization. If any atoms are uncomfortably close and clashing, OpenMM just nudges them apart so the protein settles into its most relaxed and stable state. This stark contrast is exactly why this matters. By turning $1 million, multi-year biological hurdle into practically instantaneous computation, AlphaFold changed medicine forever. What used to be a massive bottleneck for researchers, whether they were designing new drugs, understanding diseases, or engineering enzymes to eat plastic, is now essentially a free, instant database lookup.
I'll leave you with this thought to ponder. The ability to predict these shapes instantly is just the beginning of a completely new era in science. With the 3D structures of almost all known proteins now mapped out, the blueprint of life is finally readable. The only question now is, what are we going to build with it?